Once again, this is the mRNA. As you know, it is transcribed by the DNA. The codogens have made these codons. Now, triplet codon. Remember, on the mRNA, there is always a start code, which is always and always AUG, which is called as the start codon. mRNA also has a stop codon. Stop codons are always at the end. They are usually one out of the three. Now, there are three stop codons. One is UAA, UAG, and UGA. And here it is UGA. The other two are also stop codons. Now, this entire unit, suppose it codes for a specific protein, and its RNA is going to translation. Now, there is specific tRNA which carry an anticodon for this codon on mRNA. Like for example, if the codon is on mRNA is AUG, what or which tRNA should bring the specific amino acid for this mRNA? If the codon is AUG, the anticodon is going to be complementary to it, that is UAC. Similarly, for UCU, the anticodon is going to be AGA, CCU, UUC. No anticodon because no amino acid is specified for this particular stop codon, hence nothing should appear. And as I said, each tRNA is carrying a specific amino acid. Like for example, UAC. The tRNA consisting of UAC anticodon carries methionine. AG is for serine. The tRNA with CCU anticodon is glycine. The UUC is lysine, and nothing for the tRNA with anticodon. But remember, genetic code is always based on mRNA codes, not on anticodes. So genetic code hai, it is not genetic anticode. So always when you read the genetic code, you have to read the mRNA codes, not the tRNA anticodons. But every tRNA is carrying a specific amino acid based on the anticodon present. So tRNA is very specific anticodons and a very specific amino acid. Now, once transcription is done, the transcription is done in the nucleus. And you can see in this cell, the nucleus has the mRNA ready for the next step. The next step is going to be translation. Now, for doing translation, it is going to happen in the cytoplasm. In the cytoplasm, the translation takes place in the protein factories called as ribosomes. Now here I have labeled one ribosome there, which is consisting of two parts. One is labeled as L for larger subunit and S for smaller subunit. The mRNA is supposed to travel from the nucleus to the cytoplasm in the ribosome for the process of translation. Now once inside the ribosome, the translation will start. The entire process of translation involves three steps. The first step is called as activation of amino acids. Now, before translation starts, the amino acids are to be activated. Like if you remember, in replication also the nucleotides are to be activated. Here also amino acids are activated with the help of ATPs with a very specific enzyme called as aminoacyl tRNA synthetase. In the presence of magnesium ions, they make aminoacyl AMP enzyme complex and plus 2 phosphates are released. So once the amino acids are activated, the activated forms are called as aminoacyls. Now, in the second step, the aminoacyls are to be attached to specific tRNAs. Here again, the specific enzyme complex present with the aminoacyl identifies the specific tRNA, and now they are a complex of aminoacyl tRNA. Now, this is the very important complex formed aminoacyl tRNA complex. Sorry. Now, this is the very important complex formed aminoacyl tRNA complex, very, very specific, which is going to now get ready for the translation process. And third step is formation of polypeptide chain, the final step, which is going to involve three sub-steps: to start, to elongate, and to stop. Start the process of polypeptide chain going to start with AUG and end with any one of the three stop codons UAA, UAG, and UGA. So let's start with the first step. Before that, also understand the ribosome structure. Ribosome structure, you know that in prokaryotes and eukaryotes, there's a small difference. In prokaryotes, the smaller subunit is 30 Swedberg and larger subunit is 50 Swedbergs. And in eukaryotes, the smaller subunit is 40 Swedberg and larger subunit is 60 Swedberg. You notice in the diagram, the larger subunits have some three types of sites on them. The first site is called as the A site, the second site is called as the P site, and the third site is called as the E site. So here I'm calling the A site as the first site, then P, and then E. So the sequence will be always A, P, and E. Now during translation, both the subunits have come together and now they will be called as, in eukaryotes, 60 Swedberg and 40 Swedberg. When it comes together, it is 80 Swedbergs. And in prokaryotes, when 50 Swedberg and 30 Swedberg come together, it is 70 Swedberg. So this is the basic structure of ribosomes. Now mRNA is supposed to bind to the smaller subunit. Remember that. And the tRNAs are supposed to bind to the larger subunit in this manner. They will always enter from the A site. But there are some exceptions we will discuss later. So remember, the smaller subunit for binding of mRNA and the larger subunit is for the tRNA. So what you see in the diagram is a tRNA which having a specific anticodon and a specific amino acid. Now, this is the mRNA. You start the process binding to the smaller subunit of the ribosome. And the mRNA is always bound from the 5' end, having the G cap over there and ends with the 3' end. As you can see, you know that the mRNA starts translation from a specific codon. It is always the start codon. The initiation happens only at the start codon. So at AUG, as per the genetic code, the amino acid methionine is supposed to be transferred from the amino acid pool to the ribosomes. So a very specific tRNA which carrying the anticodon of AUG having methionine is the amino acid is supposed to bring the methionine amino acid here. And soon, immediately, the larger subunit are going to join the smaller subunit. As you can see, the first amino acid tRNA complex has entered from the P site. Now, second onwards, all are going to enter from the A site. Remember, the A site is named after the aminoacyl tRNA complexes because they enter from the A site. P site is named as P site because it is responsible for making the peptide bond that is because of the enzyme peptidyl transferase. And E site is responsible for the exit of the tRNA after transfer of amino acid. The second code is UCU. A specific tRNA is supposed to carry a specific amino acid as per the genetic code with a specific anticodon. As you can see, the UCA anticodon is AGA and it's got serine to it. So entry from A. Now P site is supposed to make a bond between the two amino acids in the presence of an enzyme peptidyl transferase. Now here is the bond between the two amino acids. Once the amino acids are transferred, the first tRNA is supposed to take exit. So as I said, entry from A, P will make the bonds and E will be for exit. Now, to read the next code after UCU, the next code is GGA. To read the next code, the ribosome is supposed to start shifting towards the next code. This movement is called as translocation of the ribosome. Now this process is also going to be called as elongation. Now initiation is done, the amino acid synthesis, protein synthesis has initiated because of EUG. Now elongation is going to continue. Now elongation may, you see the ribosome is shifting ahead and the next A site, a new amino acid will enter for GGA codon, and it is also going to carry a specific amino acid. A specific amino acid glycine, when the anticodon is CCU, is on the A site. Now once again, as expected, the P site is supposed to make the bond between serine and glycine, the peptide bond, and the chain is getting elongated. Again, the exit of the second tRNA which came, and movement of the ribosome translocating to the next code. So elongation has started, another new tRNA, aminoacyl tRNA complex has come, and it is going to bring the specific amino acid, जैसे कि यहाँ पर it is lysine for AAG with the specific anticodon. Again, P will be for the peptide bond formation and E will be for exit of the tRNA. Again, ribosome will move to the next step. Again, a new tRNA amino acid is come. Here it is serine. Again, P is for the peptide bond. Again, exit. Like that, you see that here is the chain of amino acids getting elongated. Now, how does it stop? The termination. You remember, the termination occurs at the one of the three stop codons. It can be UAA or UAG or UGA. As you can see on this mRNA, the next code is actually a stop code. Now, what happens when there is a stop code? Now, the ribosome moves, but now this time there is not going to be any tRNA, but there are going to be some release factors. Because AUG does not code for any amino acid, the release factors are going to help release of this peptide chain, and it will be isolated outside the ribosome. The tRNA will take exits, and this is how the peptide chain is formed. Now you'll wonder what should happen to the mRNA after the stop codon. Now once the stop codon is there, after that the segments there are actually called as UTRs. UTR stands for untranslated regions, which don't get translated. Now what should happen to this mRNA? This mRNA, either it is retranslated or it is degraded. And thus above the chain of amino acids is the primary protein formed, which will undergo further changes to make secondary protein to tertiary structure. So this is what is translation, making the protein from RNA. So what you notice from the entire process is that the mRNA functions to carry specific message from the DNA to the ribosome during protein synthesis. And what is the function of tRNA here? The tRNAs was responsible to bring specific amino acid from the amino acid pool to the ribosome during protein synthesis. Whereas the rRNA, that is the ribosomal RNA, is the caretaker of the factory. It is supposed to help in binding the mRNA in the ribosome during protein synthesis. The rRNA also helps in release of the tRNA after the transfer of amino acid. The rRNA is also responsible to protect the mRNA and tRNAs from the proteolytic enzymes formed. Also understand what is monosystronic and polycystronic. Now in eukaryotes, our DNAs are monosystronic, means one DNA at a time or one gene at a time produces only one protein. Whereas in bacteria, it is possible that at a time, one gene can make not more than one type of protein. So at a time, one transcription only is monosystronic. At a time, many transcriptions on a specific gene is polycystronic. Like here it is eukaryotic, that a specific segment is only making only one protein. Whereas in prokaryotic, that between one transcriptional unit, there are three proteins synthesized, hence called prokaryotic.